Okay, good morning. It's a pleasure being here. So I'm going to give you a brief overview of DNDi's Filarial portfolio. So for those who are not familiar with DNDi, DNDi stands for Drug for Neglected Diseases Initiative. It was created following the winning of the Nobel Prize of Peace by Médecins Sans Frontières in '99. And following the winning of the prize, MSF funded a working group called DNDi, and this group created DNDi in 2003 to address gaps in research for neglected tropical diseases. We've got 7 funding members, which are listed in these slides, and we have TDR, WHO TDR as a permanent observer. So what is our model? So DNDi trying to deliver therapeutic options for neglected tropical diseases and trying to address patients' needs. And for that, we have projects along the whole process, the whole pipeline of development, of drug development. We have a short-term project which aims at, for instance, registering registering drugs, extension of geographical extension, etc., or repurposing new drugs, or even long-term projects like new chemical entities. So what I'm going to focus on now is DNDi started working on filarial diseases a few years ago, and we're focusing on these 3, so loasis, onchocerciasis, and lymphatic filariasis. So there are other filarial infections, but these are the most relevant, at least the one we are focusing our efforts. So what is common on all these diseases is they have a common life cycle, and following the bite of a vector, hosts get infected with larvae, which then molt to an adult stage, and this adult stage then produce microfilari, which are not able to complete their life cycle in the host and needs to go back in the vector to complete their life cycle, becoming infective and continue spreading the disease. So what is important to understand for the rest of the talk is that in the human host, you've got 2 stages of the parasite. You've got adult worm and you've got microfilari. And this is important, especially because of the first filarial infection presented on that slide, loasis, where when you have a co-infection, the current therapeutic options become difficult to implement because the loasis has a high burden of microfilari and can cause death of the patient. So what we're aiming at here is to identify drugs targeting the adult stage of the parasite. So how do we started filling the pipeline? So we started with a repurposing approach. So we have been screening compound from several pharmaceutical companies trying to identify compound active against onchocerca parasite. The repurposing approach allowed us to identify two compounds, oxfendazole, emodepside, flubendazole, also it's a cousin of oxfendazole. And we also identified quite a number of hits and leads, which allow us to start a lead optimization program, especially with Celgene and AbbVie. And on their own, AbbVie and AWOL has been doing other lead optimization program targeting Wolbachia, which I'll come back to a minute. So what is emodepside? Emodepside is a veterinarian drug which was discovered by Astellas and licensed to Bayer, and it's on the market at the moment for treatment for deworming cats and dogs. So it's sold in combination, and the brand name is Profender or Procox. And these drugs got activity against several filarial species, including Onchocerca volvulus. And we are having a partnership with Bayer to develop this drug, which is currently in phase 1. The other one is a tylosin analog macrofilaricide called Talamarko. So this work was conducted by AWOL and AbbVie. And they've been screening several compounds aiming at targeting Wolbachia, which is a bacterium living inside some filarial species and which is essential to the viability of the worm. So along the screening, they identified tylosin, which is a drug on the market as well. It's a food additive for treatment of dogs. But this molecule is poorly bioavailable, so it wasn't really progressible into humans. So what AbbVie and AWOL's been doing is a lead optimization program to improve the ADME properties and maintaining the activity against the Wolbachia. And the molecule on the top was selected to be our preclinical candidate. And now this molecule is moving in phase 1 this year. So that's also another interesting and promising treatment that could be developed. The last one is oxfendazole. So oxfendazole is also a known anti-helminthic drug. There are several brands and several drugs on the market. So this molecule is very attractive because it's already in phase 1. It has been brought in phase 1 by a group called the Oxfendazole Development Group, and they're looking at treatment of neurocercosis and tinea. So our aim is to access the data of their phase 1 and extend the indication and trying to develop it for a filarial indication. So we're aiming to start a phase 1 in the coming maybe next year. Lastly, what I'm going to present, this is a bit busy slide, but I'm trying to break it down for you. So the last part is the discovery part. So as I mentioned, we have identified quite a number of interesting leads from our original screen, and we're collaborating with AbbVie and Celgene to optimize this molecule and make them available to be a suitable macrofilaricidal treatment. So this is the organization of our consortium of partners. So on the top left, you've got the academical partners who's doing the in vitro work. So we're collaborating with Northwick Park Institute for Medical Research here in London for screening against Onchocerca parasites, and with University of Bonn and the Museum of Natural History in Paris to do the parasitology. And the pharma company AbbVie and Celgene are working also with a Chinese CRO to improve the ADME properties. And the final box down is the in vivo efficacy, which is conducted also by our academical partners. And so these give you just the structure or how the consortium working to develop new molecules. So just as an example here, I'm taking the example of the Celgene compound. We started by screening a focus set of molecules, very small number, about 160 molecules. We screened them against microfilari and identified some hits. We then rescreened the entire library against adult parasites, and we identify some compound which have selective activity against adult worm, which is extremely attractive because then you could treat people co-infected with patients co-infected with loasis. And these molecules then were tested in a murine efficacy model. So unfortunately, it's not possible to grow Onchocerca parasite in a rodent, so we're using another filarial species, which is called Lithomozoides sigmodontis, which is a filarial species growing into a rodent. And we have obtained fairly good activity, as you can see, with several compounds from distinct chemical classes, which give adulticide effect. And we repeated some of these work in a gerbil model with the same parasite. We're using flubendazole as a positive control group. And as you can see from that slide, we can obtain similar activity than flubendazole in terms of adult worm. and we also see a slow decline of the microfilariae, which can be followed in a gerbil model. So these molecules are very attractive. They're not yet ready for preclinical candidate, but we have good hope that one of these can be optimized and become another treatment. I'll just conclude the presentation by acknowledging a whole lot of people willing to collaborate on this program. There is a lot of committed groups to this research, and I think it's important to realize that for any project of that type, you need a critical mass, and this slide just highlight that. And lastly, I thank all our donors for sponsoring us.